Welcome to the Streptococcus suis session at the Lemon Conference. My name is Albert Rovira, and I'm going to be the chair of this session. This year, we have decided to include Streptococcus suis in the program at the Lemon Conference because it continues to be a big problem for farms in the United States. We have a great panel of speakers that are going to cover diagnostics, research, and control. And our first speaker is Dr. Matt Sturos. Dr. Sturos is a veterinarian. He graduated from the University of Minnesota, and he's also a veterinary pathologist who works at the diagnostic laboratory in Minnesota. Dr. Sturos is one of my colleagues, and I'm very happy to have him speaking today because he's really good at incorporating new technology into the diagnostic work that we do. He's also very good at communicating it. Dr. Sturos' presentation is called Saying Hello to Our Little Friend, Streptococcus suis Diagnostics. Go ahead, Matt.
Well, thank you for that introduction, Albert. Welcome all to our session here, wherever you're watching. Hopefully the weather is good and everybody's healthy. As Albert said, title is Saying Hello to Our Little Friend. Perhaps some of you watching have had your doors blown off by Streptococcus suis, so I thought it would be an appropriate discussion here. Or maybe it's coming for you. So we're going to talk about diagnostics, what you can do. The outline of what I'm going to discuss today is you think you have strep. So what all is involved with kind of figuring out whether or not you have a strep problem. Then we're going to talk about making sure that you actually have Streptococcus suis infection and not something else. Maybe you've got both. Once you have your diagnosis, what do we mean when we say which strep? And then the end is what to do with that information once you've obtained it. So when we think about you think you have it, we need to know what it is, and we also need to know kind of some of the typical clinical signs. So when we ask what it is, Streptococcus suis is a gram-positive cocci bacteria. Yeah, that's a lot of words, Matt. What does that mean? So cocci is referring to the shape, so it's a round bacteria. The gram-positive is referring to a special stain. So the blue that you see on the outside of the circle is representative of the peptidoglycan, which is in the cell wall. It takes up the stain, and that's where that gram-positive comes from. It looks blue underneath the microscope after it's been gram-stained. This bacteria is also encapsulated. Sometimes it's not, and so that's why it's got a plus minus on there. What do I mean by that? So on the outside of the cell wall, there's also a layer in the encapsulated variants where there's proteins, sugars on the outside of the cell that helps it evade the immune system and is important for identification as far as further subtyping. Another feature when we look at it under the microscope is it tends to form chains or kind of like strips, hence where Streptococcus comes from. It's important to realize that Streptococcus suis is part of the normal pig microbiota unless we do some intervention, C-section derived. Most every pig is going to have Streptococcus suis as a part of its normal flora. There are some times when those normal flora can overwhelm and cause disease. There's other times when there's a new introduction and the pig doesn't have the immunity to deal with it and can cause disease. Typically, what we're seeing is disease in young or maybe naive pigs. So if a particular site hasn't seen a serotype before, you may start to notice signs where they don't have much problems with their existing serotypes. It's also important to know that this is occasionally a zoonotic pathogen. There are reports of human septicemia, so there is a public health component to Streptococcus suis that we should worry about as veterinarians and public health experts. When you think about the clinical signs, there are kind of different groups of clinical signs that might be seen. There are central nervous system signs. It's important to define what you mean by central nervous system. In this case, most of the signs are due to meningitis. In this case, you're going to see incoordination. Pigs might become laterally recumbent and start paddling in the end stages. You may see occasions where the infection has spread into the joints. You might see swollen joints, and that's due to the arthritis. And those pigs will be lame. Sometimes there's non-specific. They might be a poor doer. Those pigs might have other conditions going on. They might have a PRRS infection that's complicating things. and they've developed polysericitis that they haven't died from yet, or they've also maybe they've developed a secondary bacterial pneumonia from Streptococcus suis. And then another feature that is common as far as a presentation is just sudden death. We hear all quite frequently, we've got sudden death in good pigs. You know, what are we dealing with, doc? Usually that's due to septicemia, and there's not a lot that you're going to see in those pigs when you do necropsies. So when we think about how are you going to make sure that what you're dealing with is actually strep, there's a few different parts to consider. There's case selection, there's sample selection, and then there's the diagnostics to perform on those samples. When we think about case selection, ideally you would be looking for acutely sick or potentially sudden death pigs that are hopefully freshly dead. If you're not sure the interval from death to sampling, those may not be the best pigs. And ideally we want pigs that haven't been previously treated with antibiotics. That's not a game changer as far as not being able to get you an answer, but it does help to make sure that antibiotic treatment won't interfere with some of the diagnostics that come downstream. When you think about sample selection, you've picked out 2 or 3 pigs that you think have disease, and you're gonna sample them. Ideally, I'm a pathologist. I like to get the whole pig myself. And so if you can get the whole pig to the lab, check with your lab. They may do euthanasia for you. So if you've got a live animal that's not looking good, bring it in, euthanize it, and do the necropsy there. If you've got a sudden death, just bring the whole remains in. That helps us be able to rule out other systemic infections, something that doesn't fit with what you thought the presenting complaint was. And so we like to look at the whole pig. In the event that you can't, if you have paddling pigs, a minimum you need to send is a whole head. Or if you have the capability to take the brain out on farm, fresh and formalin fixed brain. Ideally, that's not all I get, but if you have paddling pigs, you better send me some brain. If you have lameness, you can send a whole leg, so keep those joints intact so we don't contaminate them. We can do the work up here in the lab. Or if you're constrained for space, you can open those up, take joint swabs and joint tissue, save some fresh and fixed, and send those in for further diagnostics. Sudden death, again, there are many causes of sudden death, so you need to make sure that you're doing a thorough necropsy and send tissues from pretty much all major body systems, so brain, heart, lung, liver, kidney, spleen. All of those will be important to rule in or rule out Streptococcus suis infection or potentially some other cause of sudden death. When we look at how do we diagnose it here in the lab, or what should you ask your lab to do if they're looking for some instructions, the first thing is you need to actually identify the bug. Streptococcus suis is one of the nice ones in that it grows well. So this is an image of a blood agar plate with a pure culture of Streptococcus suis growing. There are small kind of whitish colonies. There's little to no hemolysis around those, and it's relatively easy to grow. If you're sending samples, send us things with lesions. So if you have serosal fibrin, so if you opened up the pig and you've got fibrin on the serosal surfaces of the heart or the pleura or in the abdominal cavity, sample that. If you've got a pig that's down and paddling, we would culture the brain, lamenesses, joints, systemic tissues typically is liver. There are PCRs available, and those would be helpful for cases where you don't have fresh tissues, so you can't culture that. Maybe if you've got a formalin fixed paraffin embedded block. This PCR doesn't help you as far as further characterization. Typically, there are some specific serotype type PCRs or virulence type PCRs that may be helpful. Typically, that's not used in the diagnostic stream. And then the other part of because we mentioned that the bug is present in pretty much every pig, just the identification of the bug is insufficient in and of itself to consider that you have Strep suis associated disease. So you do need to identify and verify a tissue lesion. That could be through your gross exam, your postmortem examination necropsy, or it could be on histopathology. And so an example of a gross lesion that you might see, This is the thoracic pluck from a pig. The heart is in the center and the lungs are kind of off to the left. On the heart surface, there is delicate yellowish fibers. That's fibrin. So this is a fibrinous pericarditis or epicarditis. And then when we look out at the lungs, the lung here is red, and this is consistent with kind of a consolidated type lung. So this would be indicating there's probably a bacterial bronchopneumonia going on there. And then when we look at the margin of the lung lobes, there's also some tan material. This is fibrin, so there's a fibrinous pleuritis here as well. It's important to realize that this lesion is not pathognomonic for Streptococcus suis. Most people, when they open this up, they would think maybe Glasserella parasuis, also known as Haemophilus parasuis, Glasser's disease. So there are a number of bacteria that can cause similar lesions, so it's important that you ID the lesion and you ID the bug. This is a photomicrograph of a slide of brain. So meningitis can sometimes be subtle. So if you don't feel like you're recognizing the meningitis grossly, don't be alarmed. Sometimes it's hard to see. There's a little bit of purulent material along the meninges. But what this image is showing is this pink, all this pink material has normal brain. So this is the cerebral cortex. But out on the margins here, there's kind of like a bluish black frosting on the outside. These are all inflammatory cells. We can see some inflammatory cells infiltrating around the blood vessels as well. So when we go look at that at higher magnification, all of that blue or dark kind of colored is inflammatory cells. Many of these have lobulated nuclei, so the majority of these cells are neutrophils. And so this is the meninges right here, markedly expanded by all those infiltrating neutrophils. We also see those coming here along the blood vessels, trying to get out into the brain proper. So this is suppurative meningoencephalitis. There's a little bit coming here into the brain, and again, you may not appreciate it grossly as well as this histo slide is showing. So after you've identified it, you say this is Streptococcus suis. I'm good to go. The question becomes, well, is this the same as I had before? Is this a new one? And so the question becomes which strep. So the next section here, we're going to talk about Streptococcus suis subtyping and just touch briefly on some of the virulence associated factors. The subtyping, kind of the classical or time-honored way of subtyping bacteria, and particularly Strep suis, would be serotype. And so serotyping is antigenic categorization of the capsule. So if we go back and think about those three bacteria I showed in the beginning, they've got this yellow capsule on the outside. There are specific antibodies that have been made for well-characterized strains, and that those antibodies are what are going to be used to tag and figure out what these three are. And so in this example, the first strain, the first isolate, is identified by this black antibody. The second one, this one might be identified by this orange antibody. The third one is identified by the blue antibody. And so these would all have different serotypes. And one of the problems is usually this is performed by a reference lab. This is highly input intensive, so you need to make the antibody, have to be characterized, and then so usually this is performed by a reference laboratory. Sometimes we'll send isolates off to the reference laboratory, ask for a serotype, and they say it's not typable. So this one up here, this strain up in the upper right, is missing the capsule, and so this one right here, there's no antibodies binding to it. And so when we look back, we would say, okay, well, this one is a serotype 1. We have a serotype 1/2, a serotype 14. But this one right here is not serotypable because it lost its capsule, or it never had one to begin with. And so this creates a problem in that we're not able to fully characterize every strain just by serotyping. So that brings up the next version of subtyping, and this one is going based on the genotypic characterization. Sequence type, you may have heard that before. This is typically multi-locus sequence typing, or MLST. This is used in many bacterial species based on housekeeping genes that are conserved throughout all of the strains. And so in most cases, it's 5 to 7 genes. Streptococcus suis algorithm uses 7 genes. And this is important to point out that it's based on the allele, so you have to have the exact allele to match with that particular locus in order to be categorized as you know, the allele number 1. And then it's the constellation of those different numbers that ultimately ends up with a sequence type. So if we take those same 4 strains of Strep suis that we were trying to characterize, we know we have serotypes on 3 of them. When we start to look at the sequence type, we're going to delve into the genetics. It's important to kind of think about this as the serotyping is antigenic. That is something that's on the outside. It can be characterized by the immune system. Sequence type is genetic. It may be somewhat related to the antigenic portion, but it's not necessarily tied tightly in that respect. And so when we look at those 4, when we do the MLST typing, we might come up with a sequence type 1 for this one that's serotype 1. Maybe we get a sequence type 28 for the serotype 1/2. And again, these are all hypothetical. Sequence type 926 for our sequence type, our serotype 14. And the one that we couldn't type, that one actually comes out as a sequence type 1. So based on those housekeeping genes, this is likely highly related to this one over here, this other sequence type 1, where we wouldn't have had that information if we were waiting purely just on the serotyping. Currently, there's over 1,200 documented sequence types for Streptococcus suis, and again, that can get a bit bulky, and so oftentimes the sequence types will get lumped together into clonal complexes, and there'll be a one sequence type that's kind of been the standard for that clonal complex. So there's a clonal complex 1 that would include those related to sequence type 1. And think about it as the numbers don't necessarily, you know, 1 may be related to 2, but doesn't necessarily have to be. So it's the order in which those alleles were defined and identified. So that was looking at a subset of the genetic material. When we get into more advanced stuff, we're starting to look at whole genome analysis, and this can become much more complex. We talked about 5 to 7 genes in MLST. When we think about Streptococcus suis, it has about between 2 and 2.5 megabases or million bases that exist. So MLST typing is taking a very, very small portion of it and applying a label in that method. So when we talk about the whole genome analysis, you can do whole genome MLST. That's not something that's done typically in the diagnostic side currently. That may change as more people do whole genome sequences. So that would be a way to use the whole genome in that respect. Oftentimes when people are talking about whole genome analysis, they're looking at from one strain to the next. They may not necessarily be applying a specific label to it. And those comparing strains is often done using one method is single nucleotide polymorphism analysis, or SNP analysis. If you've heard that, what that requires is a reference sequence. So most of the time you'll select a sequence that's closely related to the one that you think you have. And then you have a sample sequence. And this sample sequence is then going to be compared. So it's aligned to make sure that it lines up with those portions of the genome. And then it looks to see where there might be differences. So in this sample sequence, I've highlighted 3 red polymorphisms that have occurred here. So in this case, there's a 3 SNP difference between the reference and your sample. One of the questions that comes up is, well, how many SNPs do I need to worry about? That has to depend somewhat on the bug that you're looking at. So again, we mentioned that the genome for Strep suis is right around 2 to 2.5 megabases. The literature suggests that the change or the random mutation that occurs, these SNPs, will occur at a rate of between 2 and maybe 20 SNPs per genome per year. And so if you're looking at an isolate from January and you're looking at an isolate from July, and there's 6 SNP differences, that could very easily be the same bug. There's just been some changes in it. If you're looking at an isolate from January of 2 years ago, and you're looking at January of this year, and you're seeing 25 to 30 SNP differences, that could still be the same bug that's been changing over time in your farm. If you're looking from 2 years difference and you're seeing 150 to 200 SNP differences, it's likely that you either didn't characterize that isolate the first time it's been on your farm, it just wasn't causing problems, or maybe you have a new introduction. So it's important to keep in mind this frame of reference for your SNPs. The other thing you can do with whole genome analysis is there are targeted PCRs that can be done for certain virulence associated factors that might become intensive if you're going to do more than 2 or 3. And so you can use the whole genome data to look for all of the known or what have been published as far as people suppose might be associated with virulence. And so that would be using a profile to say that pathogen associated factor is present or absent. And then the last thing I'm going to kind of touch on here is pangenome analysis. So the SNP that we talked about, this is looking at core or conserved parts. So the reference genome has to have what the sample sequence has in order to say, yes, it's different. So pangenome analysis is a little bit different. It looks to see whether or not certain gene content is present or absent. This is harder to define exactly what's present or absent. But for example, we have 6 isolates that I'm showing here. Everywhere that there's a blue, these thin blue lines, these would represent a gene cluster, or a segment of a genome that's present, and where you see white, that's absent. And so when you look at a pangenome analysis, all of this block to the left-hand side that's conserved, this would all be material that would be included in your core SNP because all of these isolates have that. But there might be some that's going to be missing in that analysis because that genome block isn't present in your reference sequence. And so things that look very close on a core SNP analysis may actually have discernible differences based on the pangenome. And so it's important to keep those in mind. SNP analysis is really good for epidemiological timescale type changes, but pangenome analysis will tell you if there's a certain cassette of genetic material that is present. So if we look at isolate B and isolate F, they both harbor a large chunk of genetic material here that the rest of these isolates are missing. So that would be an example of how you would say, even though they look close on a core SNP, we've got some differences here. And oftentimes these will tie up nicely. They'll mirror each other, but it's not always the case. So when we talk about the virulence associated factors or markers, they're often called markers because they've been associated with pathogenic isolates. There have been research to show that you can knock many of these out, and those knockout mutants can still cause disease. So they're not necessarily required for pathogenicity or virulence, but they are definitely associated with strains that might be pathogenic. And so there are some classical ones. These are the other thing to kind of think about is many of these are characterized in very specific subsets of bacteria. So it might be serotype 2. Well, serotype 2 may not be highly prevalent in your farm or your geographical region. So when I talked about the capsule, the capsular polysaccharide or CPS genes, these are very well known to be a way that Streptococcus suis can evade or trick the immune system, and so it's a definite virulence factor. There are some others that, again, as I mentioned, you can knock out, and these isolates can still cause disease. One of those would be suolysin. That's a hemolysin. You'll see it denoted as SLY. The miraminidase release protein, MRP, and the extracellular factor, or EPF, or you might see it as EF. Those are all kind of classical ones that have been identified as associated with virulence. When you look through the literature, there are many other virulence genes that have been identified, been proposed, but they're often limited as far as the scope. So the applicability of many of those is somewhat subjective. So one of our graduate students here at the University of Minnesota, April Estrada, has been working on Streptococcus suis for a while. She's published on kind of the association of sequence type with pathogenicity, looking at a spectrum of bacteria that have been isolated here in North America over the past few years. And she's identified several, putative genes or genetic chunks that are associated with or predictive of a pathotype. And so we've started to include those as a way for folks to try to classify their strep suis. And so now, once you have all this data, the question is, what do we do with it? And so, what do you do with the subtyping and the virulence information? And then, what do you do with the isolate? You grew it. We did a sequence on it. Is there anything else that you might be able to do? Why should you isolate? So this is just a discussion of what you might be able to do with the subtyping and virulence information. This is an example of a condensed client chart that shows the core SNP. This is showing kind of the stuff that's held together that all the isolates might share. It shows a sequence type if it's available. It has a predicted serotype, so this isn't actually sending it to the reference lab to have antibodies stuck on it. It's looking at the genetic portion to say, is it likely to be this serotype? And then there's some putative virulence genes. So these CPS genes are capsular polysaccharide genes. As I mentioned, EPF, MRP, and SLY are all kind of classical variants of virulent genes. And then we've got some pangenome. These are those putative genes that have been associated with, we don't know necessarily what they are, but are highly predictive of pathogenic type in a pretty extensive data set. So pangene C, D, and H would be examples of that. So this can be pretty busy. How do you know what to do with it? And so it helps if you have a question to ask. So in this case, I've highlighted isolate F. The question that might be asked here is we submitted a brain swab. I couldn't get the brain out. we were rushed, whatever it might be. I couldn't afford to send the whole brain, but I could send the swab. And so the question would be, is the strep suis a problem, or could it be a problem? Is it something that I've seen on my farm before? And so in this case, this is a sequence type 28. When we look at the farm dendrogram, well, there's been at least 2 other sequence type 28s isolated from this farm previously, so or this flow. So we've got epicarditis and a heart valve, so there was valvular endocarditis. So there's sequence type 28 on the farm, and it seems to be causing problems. Then you look at the serotype, yep, it's a 1/2. And then when we look across at the different putative virulence genes, we see that this particular isolate contains many of the capsular polysaccharide genes, as well as those predictive C, D, and H pan genes that have been identified. So this, even though you don't have the brain to pin it down, it does have a profile that would fit with a pathogenic type, and you have other farm isolates here. So this would be, the question is, maybe you haven't started a vaccine. You would say, well, maybe this might be an option for starting, because I've been seeing problems in multiple pigs. Another question that might be asked is, I wanted to find strep suis because I wanted to create an autogenous vaccine. I sent you ahead and you grew strep suis out of the brain. I want to send that, doc. Well, the pathologist says, well, this one doesn't have any meningitis. So you could sequence that, take a look at it, and see. When we look at it, we weren't able to determine a sequence type. So that means that whatever those alleles are, they aren't in the database. The predicted serotype, we couldn't predict the serotype. And when we look at the virulence genes, most of them are absent. So this would be an example of, this is likely a commensal strain that happened to get carried into the brain when we sampled it. And so you don't want to change your vaccine for this one. So those would be examples of how you might use that. What can you do with the isolate? I do recommend doing antimicrobial susceptibility just to make sure, because we talked about it's gram-positive. It has a peptidoglycan wall. Most strains are susceptible to penicillin, which is the green line here, ceftiofur, which is orange, and ampicillin, which are red, historically and currently. Most of the strep suis isolates will harbor tetracycline resistance, so that's oxytetracycline and chlorotetracycline down here, just to show you that very few isolates are going to show any susceptibility to the tetracyclines. The other thing you could do, if it fit with your plan, would be to create an autogenous vaccine. You would ask for that to be sent to the lab of your choice and institute a control program there. So the take-home messages here are, if you suspect Strep suis disease, you need to confirm it. So pick a acutely sick pig and send the whole pig or the appropriate samples. Confirmation, you got to get both the pathogen ID and we should have lesions. And then there are advanced diagnostic tools that are available to help you monitor and manage Streptococcus suis disease on your farm. And so, it's important to start building your database now so that you have the ability to answer some of your questions later. So I do need to acknowledge the VDL staff for all that they do. Dr. Gephardt, April, Vikash, and Sunil have all been important in getting this pipeline and analysis up and running. So with that, I'll turn it over to Albert. If anybody has any questions, my email is here. And I can take questions by email if need be.
Thank you, Matt. Excellent presentation. We don't have time for questions, but we will be monitoring the live chat for questions on the chat. Our next presenter is Dr. Matt Allerson. Dr. Allerson is a veterinarian. He graduated in 2009 from the University of Minnesota. And he also got his PhD working on infectious diseases here, also at the University of Minnesota in 2013. He is the research director at Holden Farms, and we are very fortunate to have him today to share his experiences because Holden Farms is one of the systems that had to deal with this, what appears to be a more virulent Streptococcus suis strain or group of strains that have been circulating in the last few years. And they've had a lot of experience, unfortunately for them, with that so far. Dr. Allerson's presentation is a Streptococcus suis control and recent field experiences. Go ahead, Matt.
Great, thanks, Albert. As Albert just shared there, my name is Matt Allerson, a veterinarian with Holden Farms based in Northfield, Minnesota. And I'd be kind of in the middle of the presentations here today, and the main goal here for this piece would be to describe some Strep suis control measures and recent field experiences. So Matt did a nice job prior explaining some of the just broad overview of Strep suis. I'll go into a little more detail here from the field perspective. As we know, Strep suis is a fairly common inhabitant of the upper respiratory tract. Although just because it's found in the upper respiratory tract does not mean that it always causes clinical disease. And as Matt described prior, that clinical picture and disease outcome can vary greatly from system to system or flow to flow. Some of the more typical pictures you may see, one would just be acute death. So you just have a nursery site in which you find a bunch of acute deaths in a short period of time. And then otherwise, kind of down the list of those clinical signs we classically think about for Strep suis being meningitis being very common, septicemia, pneumonia, and arthritis. One really big thing from a field perspective regarding Strep suis would be that generally clinical problems are often exacerbated by other outside stressors. One that listed there would probably be the most common overall would be PRRS virus infection. And you can also think through other things in the early nursery phase like influenza virus. Other stressors to think about would be just general and environmental conditions, and then various management related factors such as mixing different cell sources into a flow or into a nursery would more likely cause Strep suis clinical disease. A couple things at the bottom there that I think are also important to always think about regarding Strep suis would be that it is a potential zoonotic agent, so something to always keep in mind for farm staff and when you're conducting necropsies, that it can be a potential zoonotic agent, and then antibiotic use. And so in the nursery phase, Strep suis is likely one of the bacteria that would be a common cause for the use of antibiotics in the nursery. So anything we can do to reduce the use of antibiotics is obviously a bonus. Second, just to go into a little bit more regarding just general field experiences. And so from my perspective, I understand from a system standpoint or farm standpoint, Strep.suis will oftentimes display differently within one farm versus another. So I'll just give my general overview from my experience, clinical observations. Our most common point to see Strep.suis clinical disease would be in that early nursery phase. So anywhere 1 week to 3 weeks in the nursery would be the most common. That doesn't mean that's the only spot we would tend to see it, but this would definitely be the most common time frame that we would see Strep.suis. Our general picture that we would see most commonly would just be the acute death. And then in terms of an actual clinical presentation, the meningitis or the paddling pigs would be our most common, and then probably followed by arthritis or the swollen joints in the nursery. The additional stressors, as I mentioned in the previous slide, are a big deal. Certainly for us, any flow or farm that's affected with PRRS, we're definitely going to see more clinical disease with Strep.suis. So anything we can do to limit the number of PRRS infections that we have are going to be a bonus for Strep.suis. These are also the cases where it would kind of come outside that early nursery phase, in that oftentimes in the PRRS infections, we may see it already at the clinical picture at the sow farm. And these would also be cases where we tend to see Strep.suis infections even later nursery and early finishing, in fact. The other big ones would be environmental. So anytime we have these large temperature swings, I think we tend to see more just bacterial challenges in general, like Strep.suis. Strep.suis also in a very humid environment, when we're unable to get the humidity levels to where we would like them to be. I think we tend to also see more Strep.suis infections. And then the last one there for us would be any mixed source sow flow that we would have. I think anytime you mix multiple sow farms together, you're going to mix different bugs. And so I think in that case, we generally see more Strep.suis cases there. Overall, I've just, not to touch on it a lot here, but I've just included, this would just be from our own internal, when we submit our samples. Matt covered this prior, our antibiotic susceptibility profile. So it's always something good to keep an eye on for Strep.suis. Over time for us, it hasn't really changed a lot. And the one good thing in general with Strep.suis, I'd say over time, has been that in general, our response to antibiotics has been fairly good to control clinical disease. The one thing that does tend to happen is that we may control the clinical bout of disease, but then it may come back. And so generally, we don't have an issue taking down the clinical picture at that point in time, but it may come back. And then the last thing there is we also do see just variation from flow to flow and sow source to sow source across the system. So that's just a general kind of picture of what I would see, and I appreciate that probably varies from system to system. So now I'm going to go into a little bit more on the epidemiology and kind of how we look at Strep.suis overall, and I'll touch on a few of the points that Matt already covered on the whole genome sequencing and further analysis. So step one, which Matt covered prior, by far the most important, is it's where everything starts, is the actual field sample collection. And so as a system, we would tend to collect higher level of tissue samples from our sites in order to understand some of these disease challenges we have with Strep.suis and other diseases. The second step there would be getting that isolate of Strep.suis from a systemic site, and then having lesions. So we would classify those as important cases where we find Strep.suis in those systemic sites. We then kind of go through and pick out the isolates we feel are, more important. So they need to meet that criteria that they're in a systemic site and we have lesions. And then it's kind of just based on what sites we want to go a little bit further on. And then we'll do have started now to do the whole genome sequencing and the further analysis that we will get into. We'll then compare the information we get back for our vaccine selection, or potentially for flow determination, or just kind of understanding things that may be happening differently in certain flows. And then I think the one really nice thing that we've kind of seen from this is that then it's just an easy way over time to continue building a sequence library and comparing things over time. So just kind of a really brief snapshot. Strep would be a common cause, as I described before, of nursery disease, or of disease in nursery pigs within Holden farms. Just looking back for the last year period, of close to 400 tissue or necropsy submissions that we would have to a veterinary diagnostic lab, 35% of those, and that's without taking out finishing cases or sow cases. This would include every case that's there. 35% of those had a Strep suis isolation from outside of the respiratory tract. Of those, and kind of indicating the importance of collecting that whole set of tissue, liver was the most common isolation location, being a systemic site, with 88 of those 138 cases, so 64% of those would have had an isolate out of the liver. And so the major take home there is that in general, in the nursery phase, it's not, in clinical disease, it's not too difficult for us to find Strep suis as a causative agent. And therefore, that's where we are able to get our large pool of isolates to use to compare for antibiotic susceptibilities and vaccine selection. So you saw a chart very similar to this in Matt's presentation prior, and I'm going to skip the steps here of going into the detail because that's his expertise and not mine. So I'm just going to go through kind of how we would use this currently from a system approach. And so I have to give courtesy here to the Minnesota Veterinary Diagnostic Lab for helping us work through this and understand this. And so Matt already had a very similar chart, and these would just be certain Strep suis isolates that we have picked to go further and have whole genome sequencing and to look at some of these virulence genes. And so just a quick snapshot, as Matt described it earlier, we have all of our cases here, just kind of a description of what the clinical picture was, the farm here covered up, and then we have our sequence type, serotype, and then some of the virulence genes with a check mark if those were found. And so just really quickly how we would use this information, it was nice initially for us to see as we selected these isolates from a lot of different farms and different flows, and it was really nice initially to see that we have kind of these two really big clusters here, which I've just highlighted A and B. So these first two big groups here, we have a lot of very similar isolates across multiple farms. So that was nice for us to see in terms of a vaccine selection. The second here, we have C here, which we did determine that this is likely a virulent strain, and so that one became a little more challenging. Is do we use kind of that one lone isolate, and we probably need to look a little bit further to see if that one does show up more in the future. Then the one really nice part, which hopefully helps us here for the future, is this category D here. We were able to just determine that those are likely just opportunistic type streps that we found in the tissue there. So those would be ones that we would likely not pick for vaccine selection and really not pay much attention to. And so kind of to go a little further into that, how we've used this approach here is that I think it's really allowed us first to better identify those Strep suis strains that may be virulent or important for our system, and primarily for vaccine selection. So we were really quickly able to kind of pin it down to two bigger clusters there of Strep suis that we would have in our system. The second would be to kind of understand the relatedness of these strains across our different flows and farms. The third is that it also allows us to select unique isolates, so we're not kind of doubling up on different strains if we choose to go with, like, for example, a multivalent vaccine. And then the last one here, which I think is really important, is just we can keep adding now to this library, and it's really a nice, easy way to look at these isolates over time to see if we have new isolates in the future, or if they changed, or if some of these change over time. So to move on into that, kind of a nice step that sets up the first prevention control measure here, which for Strep suis would be vaccination, and in this case, autogenous vaccination for Strep suis. And then some of the others I'll go through, won't go through really much more into detail, but are, as we've talked about, really important for Strep suis prevention or control. And that would be to minimize other stress factors. The one we mentioned earlier, PRRS virus being by far the biggest, timely treatment with antibiotics, control of the environment. And then one we may not think about quite as much is just from the beginning, the sow farm. So day one care, colostrum intake, and gilt acclimation. So the first one I'll just kind of spend more time on would be the autogenous vaccine or vaccination approach. I'd say the one downside, not only with Strep suis, but with other just autogenous vaccines or bacterial type autogenous vaccines, is that they can be really difficult to assess or to see if they have an impact in a system. So I just pulled out a few examples here. One more recent one, Scott D. and then the Pipestone group, some really nice work there on an autogenous vaccine that in a nice experimental challenge model was able to lower mortality and reduce clinical scores. So a more recent piece of data there that's really nice to see. And then when you look back, there are a variety out there, and I just picked out a few here that did show that vaccination could potentially help with lowering nursery mortality, and that it could potentially provide protective immunity. So there are some pieces out there in the literature that would show that Strep suis vaccines may have some potential. I'll just share kind of an example of one approach we've taken to look at different field vaccines, and specifically an autogenous vaccine. There's really no one right way to evaluate these in the system, and it can be very difficult to do. The one example we have here is we just chose to allot individual pigs within litter at the sow farm to different autogenous vaccine groups. And then we sent those off to a nursery in different batches or cohorts. We mixed them within pen in the nursery, and then therefore, when they moved to the finisher, the same thing, mixed them within pen, and then followed them as individual pigs. So these were outside of our traditional research type barns, just in normal commercial barns. In this specific trial, we had 8 different batches or cohorts from a sow farm, and we just followed these over time. So they went to different nurseries and different finishers. A little more detail in this example specifically, we had 4 different vaccine treatments. So we had a control in which we had no autogenous vaccine present, and then autogenous vaccines A and B for this specific presentation would have had a Strep suis as part of that vaccine, and then autogenous vaccine C was for a different agent. So kind of just a variety of autogenous vaccines, A and B being those that would have had Strep suis present. Then we looked at this in 8 different groups over time. So just the depiction over there, they went to 8 different nurseries, 8 different finishers. We were able to get a good different disease pressures across these groups. Some of these broke with PRRS, some did not. So it was a little more representative to what we would see across our whole system versus just using one site. Within this, we just individually identified 600 pigs per batch or per group from the sow farm. Did that 8 times, left us with 4,800 pigs total in this trial with the 4 treatments, and 1,200 pigs per treatment group. Really what we looked at over time, again, we individually identified these pigs at the sow farm level. And so our weights, pig weight was our main variable, and then mortality. Due to the fact of how we did this, we weren't able to do anything with feed conversion. The individual pig weights were at allotment, at nursery exit, and then in the finishing phase prior to the first sale of that group. And then lastly, just by counts, we looked at mortality. And so to set that up, unfortunately, in this case, we were not able to detect any significant differences between the treatment groups in the things that we looked at: pig weight, daily gain, or mortality. And so, as stated before, these types of trials can be really labor intensive and difficult to do and assess across the system. And in this case, we weren't able to find a significant benefit, but some of the other research I shared prior, I think, is promising that maybe here for the future we have some better technology and things that will provide autogenous vaccines that may be beneficial. Just to kind of share some of the data here below, I highlighted kind of a few of the more important ones. We would think with these autogenous vaccines that nursery mortality would be a very important variable. In this case, we did not see a significant difference across the different vaccine groups, with in fact numerically the control being on the lower end. Same thing for overall mortality, did not see a significant difference. And then our average daily gain from allotment to finish was identical across the different groups. So unfortunately, in this case, we were not able to detect any major significant differences in this autogenous vaccine trial. So then to move on after, so vaccination being one of the bigger tools that is out there and has potential for Strep-suis control. The next one just being general prevention measures. And so I've just got a couple basic ones here. One big one, again, for Strep-suis, we'll keep reiterating, is just air quality and environment at each visit. In that early nursery phase, we really have to have things set up perfectly to avoid some of these Strep-suis challenges. One is just, the basic walking pens, and so identifying those strep cases early, so that we're able to catch them when they are still at a better treatment success rate. Then the last ones I have there would just be day one care and colostrum intake. I think, as we know, not only for Strep-suis but other things, if we don't have adequate colostrum intake, we are going to have a lot of problems, not only Strep-suis. So this would be a big one, especially when different vaccine, if you have a vaccine program present aimed at sows, the colostrum intake piece is going to be really, really important. And then the last one there just being gilt acclimation. So getting gilts into these farms and exposed to the resident strep bugs and other bugs in these farms to acclimate them. And so that was my broad overview of a system perspective regarding Strep-suis. Hopefully was able to highlight that, as probably expected, that Strep-suis, again, is a common finding in most pigs. Doesn't always cause disease, but certainly we do tend to have a consistent health challenge with Strep-suis, specifically in our early nursery phase. And then just to point home, the three major things to think about is it's obviously a pig health, really important for pig health, and a challenge from the public health standpoint and the zoonotic potential of this bacteria. And then the last one there being antibiotic use, and anything we can do to limit the clinical impact of Strep-suis is going to lower antibiotic use in a big degree. The last two here, I think, are just more things that are promising for the future. Is that I think creating these autogenous vaccines that are effective and being able to evaluate them will become easier here for the future with some of the newer technology and methods that Matt described prior, and that I went through of how we would look at that in this presentation. And then the last piece here is just kind of similar, that just the newer diagnostic method and technologies will really better allow us to pick those strains that are important to us, those that are virulent, and then just give us a better way to look at these different isolates that we find across farms over time. And so with that, I'll finish here with the last slide. Thanks for tuning in here today, and I think at some point here you may be able to ask some questions. So thank you.
Thank you, Matt. We have time for a question. So I wrote a couple things here, and I was wondering if you could maybe expand a little bit or share what's your ideas on gilt acclimation measures that could work for Strep-suis. I know it's not an easy question, but we're seeing in other farms that they're having this Streptococcus suis problem already in the farrowing room, and we're seeing people wondering what's the best way to acclimate gilts specifically for strep. So I'm wondering if you have any thoughts to share on that.
Yep. Good question, Albert. I tried to describe our, more of our picture here. I would say we do have cases in farrowing at the sow farm level already. It wouldn't be our most common. I think the biggest ones you can think about there would be, that's where the autogenous vaccine can come into place to, you can pick what you want to expose them to and when. And then, the other piece, I think, for Strep suis, and any kind of gilt acclimation or most things is to try to get that exposure as early as you can. And so, being maybe more common that sow farms would introduce gilts at a younger age, just generally gives more opportunity to get those animals exposed at an earlier age, and hopefully not shedding as high a level when they farrow. So those would just be two things I could think of there.
Sounds good. I have another question in relation to the trial that you did. Do you think, I noticed that even the control group, the mortality wasn't very high compared to some of the numbers that we're seeing in other farms, other scenarios. Do you think in a way maybe you were unlucky with, like it happens sometimes in trials with this particular group of pigs, and they were not as affected, and maybe that's why you didn't see results?
Yep. Yeah, that's a good point there. I mean, overall in those groups, the mortality numbers there weren't alarming. And so I think that's, the challenge when you use a field type model is you really don't know what you're going to get. And so what we tried to do was replicate as best as we can what those animals would see in the field. And a lot of those groups did see PRRS and other things. The other, model that you can go down is the experimental model, but then again, you've kind of got advantages and disadvantages with both of those. Is that then in an experimental case, it's not as a natural of an exposure. So you've got, some issues in both ways. But yes, that is we didn't have time to go into that trial as part of this in great detail, but any field trial, I think that is the definite downside, is you're kind of stuck with what you get when you do it.
And one final question for you, Matt. You mentioned how common it is to isolate Strep suis. We see it all the time at the diagnostic laboratory. And could you share some of your experience on how to pick the ones that you're going to look further into them, that you're going to characterize further with whole genome sequencing? How do you decide on which ones are you going to pick?
Yep, good question. If you go back all the way to like Matt, I think, described the tissue collection part really well. So making sure that you're picking the correct pig to begin with is probably the most important to start with. So in our case, generally those are more straightforward, being meningitis is a very common clinical sign that we would see. And so those pigs are really easy to pick out, and just trying to get tissue from those pigs before you treat them. We are fortunate that we're really close to St. Paul, so it's easy for us to also get live pigs to the diagnostic lab, which Matt mentioned, and that's been really helpful for us for these bacterial isolations, especially for Parasuis as well. And then, ultimately picking the ones that we would further characterize. One thing we start with for sure is like multiplication flows would be a big one for us, just expecting that if you find it there, you're likely find that at other farms. And so kind of starting from that top piece, identifying those first, and then second would probably just be picking those flows or farms that have more severe clinical issues to further characterize. Unfortunately, in our case, when we did that, we didn't end up with 20 different groups that it broke down to. So that was kind of nice to see from our end.
Thank you, Matt. Thank you very much for your presentation. We're going to move to the last speaker of the session. Our last speaker is Maria Jose Clavijo. Dr. Clavijo is a veterinarian. She graduated in 2009 at the Universidad Central de Venezuela, and she got a PhD at the University of Minnesota in 2014. Dr. Clavijo is very busy. She holds 2 part-time positions, one with Iowa State University as a research assistant professor, and one with PAC as a health assurance veterinarian. And she has been very busy in the last years studying Streptococcus suis and doing a lot of excellent characterization of these bacteria through whole genome sequencing. So we wanted to hear what's the last research that comes from her lab. And her presentation is called Managing Streptococcus suis One Gene at a Time. Go ahead, Maria Jose.
Well, thank you very much, Albert, for the invitation to be here with you today. It's always great to be back to my alma mater, albeit virtually, but it's very nice to be able to share these discussions with you, my former mentor, PhD mentor, and then at Matt Allerson and Matt's Sturos presentation. So I think both of the previous speakers laid out a great concepts and the foundation for a lot of what I'm going to touch today. So Albert, as you mentioned, I am here representing two entities, Iowa State's Veterinary Diagnostic Lab, and then the PAC, a breeding stock company. For both of these entities, the main goal is to develop and implement highly effective surveillance tools. So today I'm going to talk to you about a multidisciplinary effort between academia, production systems, and industry that are controlling or improving and refining how we control these endemic agents. And today we're going to focus on Strep suis. So I'm not going to go into too much detail, just highlight the points of Strep suis that are important. On the Western world, this is a bug that causes significant impact on animal health, well-being, and overall the system productivity. But in Southeast Asia, for example, this is a pathogen of public health concern as it's related or it can be associated with meningitis or severe meningitis in human cases. Now, it's interesting as Matt and both Matts mentioned before, the degree of pathogenicity within this species varies. So there's always the question as how pathogenic is my strain? And I think that's a challenging aspect of really honing into and identifying the relevant strains in your herd. You know, I think a lot of effort and resources, rightly so, are going to pathogens of public health concern in terms of the whole AMR or antimicrobial resistance and youth conversation. But I think that what's lacking in this discussion is to understand and to further our knowledge on the ecology and epidemiology of the bugs for which we actually use antimicrobials for. And I think part of this initiative that we've been working on for the last 2 years now is getting to that. And then, of course, we currently don't have, or we didn't have, a swine bacterial pathogen system, surveillance system. So we get this a lot, being a diagnostic lab. You know, do we have a Strep suis problem? So my grad student pictured on your slides, Ana Paula Silva, a PhD student in my group. She wanted to look at the complex data from RVDL. And I won't bore you into too much detail, but bottom line is that compared to 2010, we've seen a significant number in cases diagnosed for Strep suis. But if you hone into 2015 to 2019, that's where we're seeing the biggest jump. So if we compare numbers compared to 2015 to 2019, we're seeing 556 cases more diagnosed in 2019 compared to 15. So that's about 2 cases a day more than what we see. So definitely, you know, there could be many reasons why we're seeing these trends, but it just highlights that Strep suis continues to challenge the health of our herds. Now let's get into the swine bacterial database. This is a project. It's being now led by my postdoc, Dr. Robert Mugavi at the VDL. And let's talk about the objective of this. So what we wanted to do with this initiative, and you know, to begin, this is an initiative driven by production systems. So a very large production system in the U.S. came to us being a breeding stock company, and said, you know, what do you guys have in terms of endemic pathogens? You know, what tools are out there that we could use to characterize that? So that very simple question led us was the inception of this project. And what we wanted to do here is just merge 3 key pieces. Okay, so we're looking at the pathogen, and when the pathogen, we're seeing everything that we know about that. Okay, what do we know about the epidemiology, about the differences in serotype? like Matt in the first talk talked about sequence types, areas of that genome and of that species that are relevant for clinical relevance, okay? Or to understand the clinical relevance of a strain. Then we merge clinical information. And I think this has been, going back to my initial comment, the critical point of this whole project is that we've had that input from not only the isolates that the production systems and the practitioners provide, but it's also the clinical information, what we call metadata surrounding that isolate. So it's not easy to be able to gather into one database all of these key pieces, right? But I just want to highlight and really appreciate all of the input, the clinical input that we obtain from the production systems that are using this initiative or this tool. So in that case, we have the flow, the age, the pathology report, sample type, date, etc. And then when we combine that last bucket would be the new diagnostic technology. So, we've done sequencers for PRRS, for influenza. Now with bacterial agents, it's a little bit trickier, right? They're significantly bigger, so there's a lot more to look, but it's also bioinformatically complex, right? So I think with the advancement of bioinformatics and how quickly we can now generate results, it ties perfectly into trying to combine the pathogen data, the clinical information, and these novel technologies to better refine how we control and manage these diseases. So this is a highlight of what I'm going to begin to show from now on. So this is our swine bacterial database. It's now comprised of almost 600 Strep isolates, roughly 120 at this point of Glasarella parasuis, 50 isolates of A.suis, and then we're starting to build up the Hyorinus and Hyacinovia database. Just keep in mind that we need an isolate to do these. So if there's any challenging bugs, and you all know of Glasarella, for example, or the mycoplasmas being a little bit more challenging, that complicates building your database, okay? But in brief, we have about 32 top swine production systems participating in this database. And I think that's another key that I wanted to highlight. It's very useful to use this technology within a production system, but it's even more useful when you can not only look at the isolates and the strains that you have in your herd, but also combine that and trying to understand what's out there. So that's what we're trying to attempt at Iowa State with building this database, is that when our producers or our practitioners use this service, they're able to see not only their isolates or their strains from their flows, but also see what's out there. And it gives a sense of understanding maybe what's the most frequently or the most widespread set of strains. And that helps with the interpretation and, going back to Matt Allerson's point, identifying the correct strain for your herd. So this is essentially what our clients or customers at the VDL get. So we have, very well explained by Matt, won't go into it, but we have phylogeny. So essentially how one strain is related to another one. And you can obtain a phylogeny tree, just like what Matt said, either using the core genome or the pan genome. And again, the difference is just the core genome only looks at genes that all of these strains that you analyze in one time frame have, and it tries to see what the differences are. A pan genome, let's just take every single gene that all of these strains have and compare them. And what I'm showing you there is the pan genome, and again, just tells you how related one strain is to the next. Moving on, we have our serotype and sequence type. Very well explained previously. Serotype, again, one of the most frequently or the most frequently used typing tool for Strep. And there's the legend for the colors representing the different serotypes. And then to the right of that is a number, which is the sequence type, MLST. Again, a very robust, it's been available more than a decade now for Strep. And the idea of including this and leaving that in the database is that we have a lot of knowledge, a lot of science and published information about the clinical relevance or the pathological implications of different sequence types in serotypes. And that, again, combining the phylogeny, the metadata, and the rest of the areas of the genome that we look into gives us that picture and understanding. Again, the underlying goal of we want to be able to pick the right strain or identify the clinical relevant strains. Then we add that to the metadata, flow, farm, site, age, the pathology report, AMR genes, so all of what you can see there in red. So a red dot indicates that for that gene and for that isolate, that gene is present or absent. And then your virulence factors, also mentioned previously. In blue, it's what we call putative virulence factors. So there's appears to be markers that might be related to some clusters and not others, or some species related to Strep suis, but not necessarily Strep suis. So bottom line is on your right-hand side here, guys, this would be genes that we still don't really have a clear understanding about their relevance. We leave them in to understand patterns of distribution, and like Dr. Gabhardt's work at Minnesota is to identify maybe genes that can let us to figure out if a strain is pathogenic or not. Then you see some orange genes there right before the blue, and those are the ones previously referred as to like the traditional Strep suis markers, your EF, your MRP, and your SUI lysine. So right off the bat, you can see, some trends, and let's go building up to from very simple to more complex data. It's clear that there's a relationship between serotype and disease. So as you can see here, the serotypes at your left-hand side, with the exception of maybe, well, that 1-2 that's up here, should be maybe down here. But the overall, what we tend to know about serotyping is any serotype above 9 is typically not associated with disease, or typically found in healthier or commensal type strains, with the exception of a couple of serotypes here, 14 and 23, that we tend to find associated with cases. This is a slide by Dr. Dan Tucker at University of Cambridge, and you can see that shift, guys. So here you tend to see, ignore the non-typables. As you can see, a large amount of isolates are non-typable, but overall, the shape of this figure is more towards the left. And when you look at healthy populations, that figure is more to your right. So when we see, have surveillance on healthy populations, and we tend to see more serotypes above 9. Okay, so we do see some serotypes that are important for disease, but you tend to see a lot of the 10s, the 12s, 15s, 16s that are mainly associated with either commensal or opportunistic strep, and we'll talk a little bit about that. So this is data collected from our database, and a couple of points here to highlight. So see, and this is just relating serotype with MLST, so that those two are sequence type, those two putting those two together. So as you can see here, just very brief overview of what we see. Sequence type 1 is mainly associated with serotype 1 or serotype 14s. Okay, when you look at other serotypes like 4s, 5s, 7s, you tend to see a lot of variation within that. So that just tells us is that within a serotype, there can be a large amount of variation genotypically. So that's just going back to that question that focusing only on a serotype might be misleading when you're trying to identify the clinical relevance of that strain, okay? You also see a lot of pink, and a lot of just pink means that in this case, means that the MLST sequence type was not found. So when you look at that, very similar to what we saw with Dan Tucker's slide, you tend to see that a lot of the non-identified or untypable by MLST tend to be on your right. So above 10, 15s, the 16s, again, with the exception of 23 and 24. And then the other thing that I wanted to highlight all the way to your right here is that we do see a significant amount that are not typable by serotyping and non-typable or untypable by MLST. And I think both of the previous speakers hit on that, and we'll talk about what potentially does that mean for us. But it's just to highlight that within our database, close to 100 or 600 isolates, we do see a significant amount of those that are able to type either by MLST or by serotyping. Okay, so again, broad, high-level trends of what we're seeing with this data. We tend to say that your serotypes found in your, or the genotypes found in your gilts is likely the genotype that you found in your piglets. I think Matt Allerson referred to that, and I would concur with him. So I'm not saying that indirect or lateral transmission does not occur, but based on our database, we presume that most of the time, the genotypes that you will find in your gilts would be the genotype that you'll find in your downstream flow. So when you guys receive this information, there's usually three big buckets, and I like to simplify the Strep-suis conversation, and I do it this way. But in general, we see like this top area right here. Again, the tree can change, so I don't want you to take that this is always going to be the shape of the tree. But in general, three big clusters. Number one would be what we call the non-typable. So again, they're not typable by MLST, non-typable by serotype. And the other important thing, again, bringing in other areas of the genome and the value of them is that we tend to see a lot of red and little blue. So if you recall from my previous conversation or previous statement, red means AMR genes and blue means putative virulence factors. So in general, this is a hallmark of commensal or opportunistic streps, is that they tend to harbor a lot of AMR genes and then they tend to lack putative virulence factors. Going down, the next kind of group that I want to highlight is your serotype 2. It can be a 1 or it can be a 14. Again, different capsules, different serotypes, almost identical genotype. Again, to highlight the importance of looking at multiple areas of the genome. This is what we define as Eurasian genotype. It's the predominant strain that has been reported in Asia and in Europe as causing high mortality, and we're also seeing that in North America. And then finally, you see what we tend to be referred to as traditional North American genotypes. So serotype 2, again, see the difference. You can find serotype 2 in low to moderate pathogenicity, but also in high mortality cases. So serotype 2, 1-2, 5, 4, and 9. And then sequence type 28, 25, 94, 108, and 977. So let's now pick this figure apart and try and see some trends that we're seeing, okay? So this is the first cluster. So this is your cluster up here, okay, guys? This is where I'm at. Let's just highlight it and zoom in. So as you can see, what was really interesting for us is that roughly 10 to 15% of the isolates that we have in our database would fall under here. And I think I'll touch a little bit about it in the next couple of slides, but as you can see, they're not typable by MLST. They're not typable by serotyping, or very few. They don't have practically any of the orange genes, those traditional virulence factors. And then, compared to other clusters, you tend to see less abundance of virulence factors. So this is the typical, been published, typical characterization of commensal isolates. So they're highly divergent, not only within this cluster, as you can see by the branching pattern, but also compared to the other clusters that we see, or the other groups of strains that we see in our tree. And this has been done by our colleagues at University of Cambridge, Dr. Dan Tucker and Lucy Weinert. They've seen genomic or clear genomic differences between non-clinical, respiratory, and systemic isolates. So they also have seen something very interesting, is that it appears that Strep-suis or pathogenic Strep-suis has gone something called reductive evolution. So again, if you guys look at back up to our strains, they tend to reduce the amount of genes. And if you see these, let's call this the entire pathogenic cluster, you see very little red and a lot of blue. And that's exactly what these researchers, I would highly recommend this manuscript from Dr. Weinert, showing that, systemic isolates are enriched with putative virulence factors. And really, they haven't been able to find what type of genes essentially are lost in one or the other. But in general, we know that systemic isolates tend to lack those virulence-associated genes. And this is a problem. And I'm going to try and tie all this very complex, genomic information to why is it relevant to you guys in the field. Disease-associated strains in general, because they're smaller, they might be less fit in the upper respiratory airway and then be outgrown by non-disease associated strains. This is actually a very interesting study that Dr. Weilman and Dr. Tucker, both from University of Cambridge, conducted, and they saw that for every one pathogenic strain on a plate, there's 5 commensals. So it's tricky, right? When we're trying to isolate from the tissues, is it potentially, are we seeing that a lot of these commensal isolates might be the wrong strain, right? There might be the commensal strains and misinterpret the identification of the proper strains. Going back to the same concept. Now, both of these graphs are showing you serotype or sequence type by the tissues where we isolated. And I just want you to focus on 2. When we see this non-typable, either by serotype or MLST, you tend to see that tonsil swabs, for example, are essentially seen in this cluster. I'm not saying with this that tonsil swabs or even lungs, as I'm showing you here, lung samples might not be a good sample. My opinion, they're not the ideal sample to figure out or to really hit on the one that's relevant for your farm, because those sample types might also come with commensal organisms. You're trying to culture and find a colony. It's outgrown, you don't get the pathogenic one. So I think that's kind of the highlight that I wanted to show with this. And you can have the fanciest, highest, best technology and minds around this, but if you put trash in, you'll still get trash out. And I think this is the same, just zoomed in, the same cluster, commensal cluster that I showed previously. As you can see, these researchers or practitioners or veterinarians, whomever is submitting these isolates, are trying to find this bug in different brain samples or systemic samples. There's also a couple of tonsil, which makes sense that they're there. But in general, all of this gets to the question of, can we utilize samples that are less prone to contamination? In that, I think Matt hit the nail there on, improved sample collection and handling. And also repeated sampling and characterization of a flow is very important. So you want to ID those relevant strains. And then another one, I always look Dr. Rosso's assessment on the importance of going back to traditional tools. NGS is like the fancy new thing and everybody wants to play with that toy, but it has to go back and tie that with histopathology and the importance of proper interpretation and selection of your pigs to be necropsied. So this is a very bright and talented pathologist from the VDL, Dr. Bailey Arruda, and she kind of showed me this information when she was reviewing diagnostic data. So when she looked at 2018, this was back then, she found that 38% of cases in which Strep-suis was isolated from the brain had no concurrent meningitis. So imagine this. A field vet opens up their result. They might have submitted just a brain swab, or they might have submitted a brain, but they saw no histo lesions, but high growth of strep. And they, might think that that's a relevant strain. But again, it's just to highlight the importance of having that combination of the bug at high amounts found in a lesion, and then hopefully collecting that sample in a way that all the way from sample collection at the farm to what happens in the necropsy floor, and then at BACT, we're able to pick the right colony. So there might be, several ways within that process that you might end up picking, again, going back to the commensal problem that we had before, picking that right strain. So I won't go into this. I think Matt really summarized it well. Lung and tonsil, just beware that commensals outgrow pathogenic strains, and then each organ should be bagged individually. So I concur with Matt sending, and Bailey would too, or our pathologist at Iowa State would too, saying that whole pigs are the ideal sample. Just leave it to them to potentially, if there's going to be some contamination, let it be the lab. Okay, so we talked about the first cluster, right? The commensal cluster. Now we move on to the next, and I'm just going to bulk in all of these in, okay? mostly where you see this orange pop up. So most isolates associated with disease, our database would be serotypes 1, 2, 14, and 1-2, and then sequence type 1, 25, and 28. Important to highlight, 30% of all of our isolates are serotype 1 or 14 sequence type 1. So this is clearly, in our database, one of the most important strains out there in the field. And if this was my farm and I found this strain in my herd, that would be the one that I would target for my vaccine production, okay? So less common isolates that we see or strains that we see would be serotype 7s, 3s, 4s, 5, and 23s. What we tend to see with those isolates is that absolutely we could see them as a primary associated with primary strep, where you essentially see no other co-infections, or you see just a very clear case of Strep.suis. But we also tend to see a lot of these, especially down here, the 4, the 5s, the 9s, this cluster down here. We tend to see it a lot with polymicrobial cases, okay? So I would say serotype 2, 28, sequence type 1 or serotype 1, these would be your in our database, the most frequently associated genotypes. And again, to highlight here, you can see how these genes, or you can see how these strains have a lot more blue and orange and a lot less red compared to the top strains. So again, combining all of that information leads to proper definition of what a relevant strain might be. And then finally, most have an assigned serotype and sequence type. So again, very big, again, trends of what we see with this database is that overall, and I just brought 2 examples, but we've seen this with sequence type 1, serotype 1, and the traditional North American ones, 22, serotype 2 and sequence type 25 and 28. You tend to see that regardless of year, you can see here 2017, 18, this is the same flow, 17, 18, 19, all of these cases would be the exact same strain. As you can see, at least at the genome, are very, very similar. Same serotype, same sequence type. You find that repeatedly over time in a system. Similar down here with this sequence type or serotype 23, sequence type 118. Again, almost identical at the genomic level, found over different, same system, found over different time points. And I think tying that back to how to type Strep.suis is important to figure out the following. Not all pathogenic Strep.suis are created equal. It's a shade, or let's say you have variation in terms of how much disease some strains can cause. So it might be, very primary, like your sequence type 1, serotype 1 strains, or others might need a little help from other more immunocompromised situations. So a viral co-infection, or maybe some management factors that then immunocompromise the pig, and then they are able to replicate and cause disease. The key here that I wanted to bring is that it's not just useful. I mean, you can have a diagnostic tool that allows you to understand at least, is it this cluster or is it this cluster? But within their pathogenic clusters, there's a significant need to identify not only if it is or not virulent, but which one it is. So I know that's trickier, but there's really there's little relevance if you can only pick, if your PCR targeting 3, 4, whatever amount of genes can't differentiate between this cluster, or this cluster up here. All of these clusters have, from our perspective, and the information generated from the field, is that the clinical impact of those, and it's also been evidence in the literature, is that the clinical outcome is different. So if they are all being identified as the same, you are still needing to clearly type by different methods, potentially whole genome sequencing, to really understand, is it my highly pathogenic serotype 1, sequence type 1, or is it a more low pathogenicity serotype 2, sequence type 28? And I just want to highlight not only University of Cambridge, but all of the valuable work that's been done at Minnesota to get to that, that identification of those markers. The other way that we've been using this, and I think this is one of the most key takeaways, in my opinion, of the use of this database, is realizing when we work with our customers, the amount of times that the vaccine strain, while including strains that we know can cause disease, does not include the contemporary isolates actually causing disease in their herd. And this is a typical example. We had this customer that had either an opportunistic strain including in their vaccine, or even a commensal strain included, and then 2 of them. So, I know there's a lot of debate. I know there's a lot of conflicting information on the vaccine efficacy, but anecdotally, what several production systems, large production systems in the US have been telling us is that if you combine the right antigen, the right antigenic mass, and a robust adjuvant, these products will be effective. Again, anecdotal information. I know Matt just presented the case of maybe potentially an unsuccessful approach, but as you saw with other information, it appears that they can be efficacious. So again, trying to correlate your disease-associated cases with those in your vaccine is a very good use of NGS. This is another case. It's the same system. So, and this is all new information. I just got this information yesterday, and I wanted to include it. But this is the strain of Strep found in the disease-associated cases, and this is the one in their vaccine. Both of these are actually very good candidates for vaccine. But if you look at it, and if we just see the sheer frequency, the clinical presentation, and what we know from the genome of this sequence type 1, serotype 1, it's obvious that the right candidate in this case would have been to also include this strain. So one of the recommendations that we're going to give with this specific client is to say, I would add to your serotype 1-2 sequence type 28 vaccine this one up here. The other thing that we've learned is that frequent sampling of a population increases the likelihood of detecting the correct strain. Again, what we saw before is that commensals outgrow them, that you could have, they're very tiny, they might not survive the whole culture process or be outgrown by commensals. So it's important to keep sampling a population until you really detect that pathogenic strain. In this case, it's a production system that I worked with. The person had sent in several isolates or tissues from different pigs. And what we were able to see is that only in 1 out of the 10 pigs, they were able to found that pathogenic compared to 9 other pigs. So we could only find that, highly pathogenic strain in 1 out of the 9 pigs. Again, the value of multiple submissions and frequent, multiple pigs and then frequent submissions from that site to really identify that causing strain. The other thing that we learned from working with one of our customers is, if we only look at serotype, you might be misled. So in this case, this is the same system, and it had serotype 2 within the commensal cluster, within the highly pathogenic cluster, and what we consider low pathogenicity cluster. So again, if this vet is just looking at typing and it only uses serotype, it might say, oh, I have, the serotype 2, just put that in. I know that that's been associated, looking at the literature that's been associated. Well, this might not be the one really that you want to put in. You might want to put in either one of these or this one. So again, looking at multiple areas of the genome gives you that more complete picture. And then going back to the whole tissue submission and commensal versus pathogenic dynamic, what we see, and this is part of a work with Dr. Arruda and myself, if we select the right pig, acutely infected pig, non-treated, you can actually find certain strains in any tissue that you look into. So look at this, sequence type 1, serotype 1, you can find it in CSF, tonsil swabs, brain, and in other tissues. Similar down here, you can find it in cerebrospinal fluid, spleen, brain, lung, Again, so this is a little of a contradiction of what I said before, that, tonsil and lung, are of low diagnostic value. But in reality, what we see is that when you have a clear outbreak of Strep suis and you only find one strain and that's distributed across different tissues, that's a pretty good indication that that's a primary Strep suis case. Dr. Gotcha at University of Montreal tends to say if you find multiple serotypes within an outbreak, you might want to look at an underlying cause of that strep. Look at it more as a consequence rather than a cause. So this is what we tell our customers when they're going to use our service, our NGS service. And there's 4 points here that I want to highlight. Important. NGS is not something, like running an ORF gene for typing for PERS. It takes a significant amount of time, roughly 3 to 4 weeks to do the analysis. So it's better to batch all of your isolates. Take some time in the front end to accumulate all your isolates. That can be contemporary isolates, vaccine strains, retrospected isolates that you want to look into, and then submit that as one case. It's useful because then you have in one tree all of your isolates, rather than submitting over time and then having your different strains distributed in different trees. So this just facilitates a lot of the work for us, and also interpretation for you guys. When you do the submission, include the metadata that we referred before, and then you can just request NGS for Strep suis, GPS, MHR, MHS, S.suis. And then timeline is roughly between 3 to 4 weeks until you get your report back. We recommend doing this quarterly, or if you have some sort of a health status change. You all of a sudden have an increase in meningitis cases or arthritis cases, and you want to understand what's going on. Okay, breeding stock perspective, right? And I think I've touched a little bit on it, but how do we use this? So not only is this applicable for new farms or farms understanding what comes into their farm via gilts, it's also important for farms to realize that within a production system, if we have the flexibility and we have the way to control those flows, the ideal scenario is that you match your flows when you're commingling them to have the same endemic profile. So in this case, what we've done with PIC, very helpful in a service that we provide to our clients, is say, well, let's identify what you have in your flow. Let's look at what we have in our multiplier. PIC is really proud of work for this company that's transparent in this point. Let's show you what we have in our multipliers, and let's try to tie that with the best fit for your farm. So in this case, just broad level, you wouldn't want to match a source A, serotype 1-14, with a B. And likewise, if you've had this strain, you don't want to receive a new set of bugs into your herd. So again, we knew this, right? If you think about conversations that you guys have probably had when you're looking at the sources of gilts that you've received, we've also talked about it, but now we have the tools to measure it and the tools to make more informed decisions. I think we're just barely scratching at the surface of what antimicrobial resistant genes and resistome mean for Strep suis and how we can utilize at the field. But a couple of things that I wanted to say here. Our pipeline includes or compares our isolates to over 7,000 AMR genes. And what we intend this what we intend to do with this data is to correlate the resistome or those AMR genes distributed within disease-associated strains, and try and tie that with phenotypic resistance, and see if we can utilize that data to refine how we use antimicrobials. But just high level here, you can already see how adding the AMR genes leads to a more clear identification of a commensal versus a high pathogenicity. That's take home number one. Then take home number two, I think it ties back to our susceptibility data or phenotypic data that we produce at Iowa State, is that all of the pathogens, all of the isolates that are associated in our dendrogram, in our database with pathogenicity tend to have very little red. So that's just a confirmation that, while yes, there's this whole conversation about antimicrobial resistance, and there's definitely some underlying resistance in the Strep suis species, what we can see, at least with this database, is that these strains are, at the genetic level at least, do not show significant resistance to the antimicrobials that we use in the field. Again, and that just ties back to our correlation with phenotypic resistance. And that just, I think, aids in the conversation of judicious use and how we do things well in the industry and are really conscious about what types of antibiotics and how to use them. So again, just shows you here that we don't see that much resistance in the antimicrobials that we treat Strep Suis for. So just to finalize a couple of take homes here. So the ISU-VDL now offers whole genome sequencing for characterization. We've been offering this for consistently for about 1.5 years now. We've created a large, well-characterized database of isolates from the US swine herds that is shared between swine veterinarians and producers. Again, it's less of use if you can only compare what you have. I mean, definitely useful, but it's really good to have that perspective of what's out there in the field, what other systems seeing, and the frequency of certain strains. It's ongoing characterization to determine the relevance of strains, and we can track changes in spread and frequency. Dr. Gebhardt, Dr. Tucker's great research on identifying specific markers that you can do for rapid detection of a particular pathotype. Again, the focus is maybe to understand if it's pathogenic or not, but then you would need further typing to really understand which one it is. Again, we can track AMR genes and understand relevance for field control, and then the use of NGS, in our opinion, will aid in the refinement of prevention and control strategies. Again, it's when you tie this, high cutting edge technology with the clinical pathological information from that isolate, and also everything that we know about that pathogen. So I would like to finish by thanking all of my collaborators. This is definitely a group effort, and it's a group of highly motivated and multidisciplinary team. So you have Ganwu Li from our bioinformatics team, Dr. Nubia Macedo, formerly from Minnesota, now at Iowa State, Dr. Sahin, our bacteriologist, Beatriz Martinez, UC Davis, part of understanding if we can use this as in the disease bioportal, and Dr. Dan Tucker and Lucy Weiner from Cambridge, trying to get to that, the holy grail of Strep Suis research and identifying global markers for disease. And of course, the king of Strep Suis, Dr. Marcelo Gocha, and my student, Dr. Robert Mugabe, just wanted to highlight their work and their effort into, at least my understanding of this whole cool project. And then finally, what makes this project is the interaction with the swine practitioners and the producers. Very grateful for all of their input, for coming up with these simple questions that challenge us at academia to try and solve, and that provide all of the isolates and the clinical information around it. And with that, I'd like to thank Albert, thank Monsi for the invitation. It's fantastic to be back here. And if you guys have any questions, feel free to reach out to me via email. Thank you.
Thank you, Maria Jose, for a very interesting presentation. Unfortunately, we don't have time for questions, but remember, we're monitoring the chat, so keep them coming that way, and we'll try to get back to you and to everyone with answers to those questions. And I just wanted to thank our 3 speakers again one more time for this very interesting session packed with information. And with that, we will conclude the Streptococcus suis session. Thank you very much.